Imagine a groundbreaking revelation that could change our understanding of how our gut interacts with our brain. Researchers have recently unveiled what they term a neurobiotic sense, a novel system that enables the brain to respond instantaneously to signals emitted by the diverse microbes residing in our gut. This discovery opens up exciting new avenues for exploring the intricate relationship between our digestive system and our mental well-being. The research, spearheaded by neuroscientists Diego Bohórquez, PhD, and M. Maya Kaelberer, PhD, from Duke University School of Medicine, and published in the prestigious journal Nature, focuses on specialized cells known as neuropods. These tiny sensor cells are located along the epithelial lining of the colon and play a crucial role in detecting a specific microbial protein. They send swift messages to the brain that can help regulate our appetite. But this is merely the tip of the iceberg. The research team posits that this neurobiotic sense could serve as a broader framework for understanding how the gut perceives microbial activity, potentially influencing various aspects of our lives, from our eating habits to our emotional states. Moreover, it raises intriguing questions about how the brain might, in turn, influence the composition of our microbiome. Bohórquez, a professor of medicine and neurobiology at Duke, expressed his curiosity about whether the body can sense microbial patterns in real time, not just as a response to immune or inflammatory triggers, but as a neural response that actively guides our behavior. This perspective is revolutionary and invites us to rethink the gut-brain connection. At the heart of this discovery is a protein called flagellin, which is an ancient component found in the flagella of bacteria, those tail-like structures that enable bacteria to swim. When we consume food, certain gut bacteria release flagellin. The neuropods detect this protein with the assistance of a receptor known as TLR5, subsequently transmitting a message through the vagus nerve, which serves as a vital communication pathway between the gut and the brain. The research team, with support from the National Institutes of Health, proposed a daring hypothesis that the presence of bacterial flagellin in the colon could activate neuropods to relay an appetite-suppressing signal to the brain. This suggests a direct influence of microbes on our eating behaviors. To test this hypothesis, the researchers conducted an experiment where they fasted mice overnight and then administered a small dose of flagellin directly into their colons. The results were telling. The mice consumed less food. In contrast, when the same experiment was performed on mice lacking the TLR5 receptor, there was no change in their eating behavior. They continued to eat and even gained weight. This finding indicates that the TLR5 pathway is essential for regulating appetite, as it allows the gut to communicate a "we'd had enough" signal to the brain. Without this receptor, the message fails to reach its destination. The study was guided by lead authors Winston Liu, MD, PhD, and Emily Orway, both graduate students in the Medical Scientist Training Program, along with postdoctoral fellow Nama Riker, PhD. Their experiments demonstrated that disrupting this communication pathway altered the eating habits of the mice, highlighting a deeper connection between gut microbes and behavioral patterns. Looking to the future, Bohórquez believes that this research will significantly benefit the scientific community by shedding light on how our behaviors are influenced by the microbial inhabitants of our gut. One promising direction for future research is to explore how different diets can alter the microbial landscape within the gut. This could be a crucial piece of the puzzle in understanding conditions such as obesity or various psychiatric disorders. But here's where it gets controversial. Could our eating habits be more influenced by the microbes in our gut than by our own conscious choices? What do you think? Join the conversation in the comments below.